These are our objectives for Chapter 13, and we will discuss each of them in the course of this video.
In the last chapter, we talked about mitosis, which can be used as a method of reproduction, particularly what is called asexual reproduction. There's still DNA that's inherited to the daughter cells in the case of single cells, or to daughter organisms in the case of, for example, this organism here. I told you I was going to tell you about Hydra. This is a freshwater organism. It lives, if you've had a freshwater aquarium, you may have had Hydra growing on it. You find it in ponds and streams and lakes. They're very tiny. They're usually transparent. Sometimes they have pigments. And they're related to coral and sea anemone and jellies, what you know as jellyfish. In this particular case, this organism is undergoing budding. It's producing offspring using mitosis. This offspring, as we said before, is going to be a clone of the parent that it came from, because there is no second parent contributing DNA. The other type of reproduction we're going to talk about, and the one that involves meiosis, is sexual reproduction. In this case, you don't get clones. Instead, you mix the genes of the parents and get a great deal of genetic diversity just in the creating the gametes, which is meiosis, and then in fertilization. Many organisms, plants for example, undergo what's called alternation of generation. That is, they spend a part of their lifetime as a diploid organism and another part of their lifetime as a haploid organism. You've walked through the forest, you've seen ferns. What you may not have known is that if you look at the underside of each of the fronds of a fern, you see these little structures called sori, where meiosis takes place. That produces the haploid spores, which fall to the ground underneath that diploid plant and form these little heart-shaped gametophytes. It's a little plant that, I know I'm drawing, it's there. It has these little roots, looks like a little heart-shaped leaf, and from that leaf, the 2N organism arises, you see right here. And because this was haploid, you're going to have a fertilization process, which is what's shown here, to get us back to the 2N organism. In our case, we have our haploid cells are contained within our diploid state. Here we have the human chromosomes. We have 46 chromosomes total. That's a diploid number. That means we have 23 pairs of chromosomes. 22 of those pairs are what are called somatic chromosomes. The final pair are the sex chromosomes. We arrange those chromosomes in order of descending length when we do what's called a karyotype analysis. Basically, you grow some cells, or you capture some cells from the body, you spin them down onto microscope slides, and then look for cells that are in the middle of mitosis. When you find those cells in mitosis, you take photographs of their chromosomes and literally cut and paste to rearrange them. So the longest of the chromosomes, and this pair has centromeres in the same place, staying the same. These are called a homologous pair. This is chromosome pair number 1. And then you decrease in size as you go through the karyotype. Now, this is the karyotype of my little green men, and it shows you actually one of the reasons why I use this model system. First of all, their diploid number is 6. They're easier to work with. And what I can illustrate in my drawings that you can't necessarily see when you look at a karyotype analysis is that each of these things is a duplicated chromosome. So on other karyotypes, perhaps not this one, you can sometimes see a shadow going down the center of these duplicated chromosomes because they're actually two sister chromatids stuck together. This is my example of a homologous pair. One came from dad, one came from mom. They're the same length, they have their centromeres in the same position, and they have the same genes at the same locus, the same location on the DNA. In this particular case, dad's gene is the allele that encodes for 4 toes. Mom's gene is the allele that codes for 3 toes. You have to have identical alleles on the sister chromatids because you made one by duplicating the other. That was DNA replication. There are 3 features of meiosis that distinguish it from mitosis. The first of them is one of the most amazing things that I know of in all biology. It's called crossing over, and it occurs between homologous pairs of chromosomes when they align with one another, really during prophase I. And what happens, this is a figure, look, it's 100 years old. It's from Thomas Hunt Morgan's, we call it the fly lab. They were working with fruit flies. I kid you not. That's a Sarah Palin joke. And they were using fruit flies as a model system to try to understand inheritance. And they were getting some really bizarre inheritance patterns that they finally attributed to the fact that what happens during early meiosis is that there is essentially, we now know, a scaffolding of sorts that is constructed between the two members of the homologous pair. And then the DNA is cut at precisely the same places on neighboring but non-sister chromatids, cut here and cut here. And then there is a swap, which is just the most bizarre thing. And then those 2 DNA pieces are reconnected now with something that came from the opposite chromosome. So here's the figure of them working out what happened. It was kind of a what the hell moment in the understanding of DNA, and I still find it a what the hell moment. So what you have here at the bottom after crossover, so what you have here is what is dad's member of the homologous pair that now has a segment of DNA that came from mom's chromosome, and then you have what was mom's member of the pair with a piece of dad's chromosome, but not the sister chromatids. The ones that were on the outside of this don't do the exchange. So it's really very strange, but it's important because it mixes our DNA, and that turns out to be good for our species.
Crossover happens during prophase of meiosis I. During metaphase I, immediately after that step, instead of the single file alignment of chromosomes that we saw in metaphase of mitosis, we see double file. We see the homologous chromosomes align with one another side by side. Eventually, those pairs of chromosomes will be pulled apart, and it's at that point where we get a haploid cell. At the end of meiosis II, we're going to have 4 haploid cells, and the reason for that is because meiosis II is like mitosis. So what you should see here is the pair, the sister chromatid for each of the what were duplicated homologous pairs. Here's the effect of crossover, and because of crossover and something we're going to call independent assortment shortly, we're going to have very different offspring. So whereas mitosis gives me 2 identical cells, it's a cloning process, meiosis does 2 things for me. It gives me half the number of chromosomes, and it gives me some what we call recombinant DNA. I'm mixing up the chromosomes themselves, and because of crossover, I've mixed up the content of the individual DNA molecules. Cell division doesn't always work properly. There are errors of meiosis and mitosis. Normally what should happen in Meiosis I, for example, is that a homologous pair is separated so that there is a duplicated chromosome in each of the cells that are entering Meiosis II. And if you can imagine that part of my X and that's the other part of my X, I've divided what were sister chromatids and now they are daughter chromosomes. Sometimes this doesn't work well. So instead of getting one cell, that shouldn't be there, that has one of each of the members, I'll get one cell that has both members of that particular chromosomal pair and another one that doesn't have any. And so when this cell goes through its second round of division and pulls those centromeres apart, it can result in having two copies, one cell having two copies of that, of each member. So what happens is that one of those cells in meiosis I will get two copies of, or both copies of a particular homologous pair. The other will get nothing. And when these cells go through meiosis II, which looks like mitosis, then one cell will have two daughter chromosomes for that particular chromosome, and the other will have the other two. These were the sister chromatids. Whereas this cell will be lacking in each of those. Now, what happens then is along comes, let's say those were ova, and along comes a sperm that has its own member of that chromosome. And in one case, I have what's called a trisomy. And the other case, I have what's called a monosomy. And we think this happens for every one of our 23 pairs of chromosomes. The most common trisomy by far is trisomy of the chromosome number 21 that produces Down syndrome. Here's an example of a monosomy where there is an X chromosome but not a Y or another X. Turner syndrome, there's some speculation because the Y chromosome is so small and has really kind of gone through some really radical changes in evolution that in some cases what is perceived clinically as Turner syndrome is not just X0 but X broken Y, if that makes sense to you, non-functional chromosome. So as I said, we think there are trisomies or monosomies of all of these different pairs. If we don't see it out in the real world, we believe it's because those non-disjunctions cause lethality long before birth. There's some that cause just really horrific birth defects that are not survivable in any case. We know that asexual reproduction is associated with a really small degree of genetic variation, mutation, and some what's called lateral gene transfer, trading genes like playing cards among bacteria, for example. That gives you genetic variation. But nothing is as profound as sexual reproduction when it comes to varying the genes of the offspring. The first reason for genetic variation is called independent assortment. And let me just go back to my little green man here. See, I have dad's chromosome here, mom's here, dad's here, mom's here. Now, how many examples are there? How many different ways are there to lay out these chromosomes so that when you go through meiosis I and meiosis II, you've seen every possible genetic combination? Well, it turns out that the way that you would do that is you would take 2 to the end. That should be a, where is it? That's 2 to the end. It's an exponent. possibilities. So in my little green men here, whoa, it should be 2 to the 3 haploid number. And so I would have 2 times 2 times 2, which is 2 times 2 is 4, times 2 is 8. So I'd have 8 possible gametes in my little green men model. Humans, on the other hand, have a haploid number of 23, and so if you, that I was writing 2 to the 3 to show you what I just did. Humans, where you take, now see, there it is again, it should be 2 to the 23rd power for humans, you get over 8 million possible genetic combinations. Now, if you're talking about making, for example, ova from an egg, you make 4 cells. So 4 out of 8, over 8 million possibilities, you're going to have some really very different gametes. Crossing over adds more genetic variation. We don't know exactly how often crossover occurs, but by some estimates, as much as 1 to 3 times per chromosome. The longer the chromosome, the more crossover events there are. So it's hard to actually give this a number. A greater impact on fertilization, because you have 1 of 8,000 possible ova being fertilized by 1 of 8 million possible sperm, and so you get more than 70 trillion possible, well, that's supposed to be zygote, genetically distinct zygotes. So that's a tremendous mix-up of genes to add genetic diversity to a population. Finally, so what? Why should we care? So we now know that different forms of a particular gene is called an allele, and those genes encode traits, toes in the case of my little green men, things like the color of Gregor Mendel's pea pods we'll see, hair color, skin color, eye color. Some are very, very complicated means of inheritance. I'm using this model system because we're going to look at it not just from the standpoint of mitosis and meiosis, but we're going to look at it to see what happens with genetics. So we're going to talk about how particular traits are inherited. So the big picture about all of this is not just who in your family looks like who else, but it's about how evolution operates on the traits that are available. It's the fact that we have mutation and multiple alleles, not just in you, but multiple alleles in a population, that allows natural selection to operate on a population. You have to have genetic variation for that to be.